In Chapter 11, we'll explore carbohydrates. So we'll go through and look at some definitions associated with carbohydrates, talk about what they do, the different functions of them, why they're important. We'll look at chirality of carbohydrates, and a lot of this goes back to what you've studied in organic chemistry in terms of the different systems, the different forms of enantiomers that are present. We'll highlight some important monosaccharides. We'll talk about how those monosaccharides are typically found as cyclic monosaccharides. And we'll look at some reactions of monosaccharides. And not listed, but we'll also talk about some important disaccharides, oligosaccharides, and polysaccharides. So larger carbohydrate structures. So what are carbohydrates? Where are they found? How do they function? What's the structure? How are they classified? These are all the things that we want to understand through our study here in this chapter. So what are these? Well, at one point, carbohydrates were thought to be not really important. They were kind of on the peripheral of cellular activity, but now it's found that they are actually recognized as a significant biomolecule. And because of that, we have a whole area of study devoted to carbohydrates, this glycobiology. And this is the study of synthesis and structure of carbohydrates and how they are attached and recognized by other molecules within biological systems. Carbohydrates are important for fuel and structural components. They are carbon-based molecules, so carbon-based structures, and they're rich in hydroxyl groups. Lots of OHs thrown in there. Carbohydrates, they have carbohydrate oxidation provides energy for cells. Carbohydrate storage in the form of glycogen provides a short-term energy reserve, so we're able to store this energy. And carbohydrates can supply carbon atoms for the synthesis of other biochemical substances. So this supply of carbon atoms, again, for synthesis of other biomolecules. So this is going to give us the carbon atoms that go into making proteins and lipids and nucleic acids. Carbohydrates also form part of the structural framework of the DNA and RNA molecules. We know that there are sugar phosphate groups, so those sugars are those carbohydrates. They're linked to lipids, and they can serve as structural components of cell membranes. And carbohydrates can be linked to proteins, and that can allow them to function in a variety of cell-to-cell and cell-to-molecule recognition processes. In terms of the general structure of carbohydrates, they have this formula, and my subscripts didn't work here, but CnH2nOn, so that's the general formula for carbohydrates. And carbohydrates are going to be polyhydroxyaldehydes or polyhydroxy ketones. Okay, so we see the aldehyde group here and the ketone group here. The polyhydroxy means that there are many of these OH groups present in the structure here. The simplest of the carbohydrates are going to be the monosaccharides. So we're going to start our study with monosaccharides. And monosaccharides are, again, they're aldehydes or ketones, and they have two or more hydroxyl groups present in their structure. Now, the simplest of these monosaccharides will be trioses. So what we have here are trioses, which are going to be 3-carbon monosaccharides. Dihydroxyacetone is a ketose, because it has a ketone within its structure. Something like glyceraldehyde is an aldose because it has an aldehyde group in the structure. So we can see here, these are all monosaccharides. They're all 1, 2, 3 carbons. They either have that ketone or aldehyde group present in their structure, and they have multiple OH groups within them. at least two OH groups in that structure. So if the 3 carbon are the trioses, we can describe the 4 carbon as being tetroses, 5 carbon as being pentose sugars, 6 carbon would be our hexose, and then 7 carbon would be our heptose sugars. In terms of the hexose sugars, glucose and fructose are going to be some key hexoses that we'll study as we move through. We can classify the different types of carbohydrates based on the general size. So if we're looking at size, the monosaccharides are going to have 1 unit. Whereas the disaccharides would have 2 units, and the polysaccharides would have many units. And one term that's left off of this list, we can also talk about oligosaccharides. And an oligosaccharide is going to have an intermediate number of units. So these are individual sugar units that in these higher structures can be brought together and polymerized. We can also classify the carbohydrates based on the number of carbon atoms that are present. So within the monosaccharides, you can look at those trioses, the pentoses, the hexoses, the heptoses. So we can classify that way as well. Carbohydrates will exist in a variety of isomeric forms. So for example, dihydroxyacetone and glyceraldehyde are considered constitutional isomers of one another. And we see that here, these constitutional isomers. And so what that means is they have the same formulas but differ in the order of the atoms that are present. Stereoisomers. These are isomers with different spatial arrangements. This is where we're going to look at that DL system. And you should have covered some of this in organic chemistry, but it just kind of relates to the different forms or where the priority lies within groups. Now the stereoisomers can be broken down further into the diastereomers and the enantiomers. Now, enantiomers are non-superimposable mirror images of one another, and diastereomers are not mirror images of one another. But within these, so one thing that we want to look at with the enantiomers, we actually end up with what's referred to as an asymmetric carbon in the center, and that asymmetric carbon will be responsible for creating the different stereoisomers that are present there. It's just a chiral carbon, and it's attached to 4 different types of atoms or groups of atoms around that particular carbon. And our diastereomers, we can have epimers or anomers broken down this way. So differing epimers differ at one of several asymmetric carbon atoms. Anomers, they differ at a new asymmetric carbon atom formed when you have a ring closure. So here we're showing 6 common monosaccharides that are present. We see D-glucose. This is the most abundant in nature. So here's our D-glucose. Most abundant in nature. It's an important source of human nutrition. Things like grapefruits and ripe fruits are a good source of D-glucose. We have D-galactose. D-galactose is milk sugar. It's synthesized in human beings. It's referred to as brain sugar. And this is the type of carbohydrate that we tend to see when we're looking at blood types. We have D-fructose. D-fructose is a ketohexose. It is the sweetest tasting of all sugars, and it's found in many fruits and honey. This D-fructose, is going to be a good dietary sugar because it has a higher sweetness, so you don't need to add as much of it to foods. Then we have D-ribose and D-deoxyribose, and these should be familiar because these are part of molecules like DNA, RNA, and ATP. And then the last shown here is D-mannose, and this is actually a sugar that's really important for glycosylation of certain proteins. So that's what's important in metabolism and some other functions. So I've already mentioned a little bit about chirality, but again, if we have that asymmetric carbon, what that means is that you have a carbon molecule that is attached to 4 different groups around it. And so we can classify molecules based on this. And this gives rise to those different systems that we can study, or the different isomers that we can study. Now, oftentimes when we're looking at biochemical molecules, we will be presented with Fischer projections. And these Fischer projections will show us this DL system. In the L system, just to differentiate between these, this OH group that's coming off of that chiral is going to be on the left-hand side, and in the R, you're going to see that on the right-hand side. So that's the differentiation here between the L and D groups when we're looking at these. Now, many of the common monosaccharides, especially those that are going to have 5 or more carbon atoms in their structure, they tend to prefer to take the cyclic form. So when these cyclic structures are going to come together, they're going to take what's referred to as either an alpha or a beta form, and this depends on how these ring structures close. So what we're looking at here is kind of the chemical basis for cyclic sugars. We're looking at these reactions that take place. You take your aldehyde and your alcohol and you form your hemiacetal. Your ketone in your alcohol forms your hemiketal. And then these things, these R groups coming off of here, you're going to end up with ring closure to form pyranose and furanose molecules or structures. So this diagram is just giving some more detail about the ring closure process. So we end up with some interaction between carbons on opposite ends of this Fischer projection. So you start with your Fischer projection. Here we're looking at D-glucose to start with. Then you look at where the OH groups are present, and you can see, this is showing with arrows, how these are going to rearrange to come together in a structure. And you end up with this ring closure. Now when we have this, we said that there were 2 ways that these rings could close. You can have the alpha form, or you can have the beta form. In the alpha form, the hydroxyl group that's attached to carbon 1 is on the opposite side of the ring as the carbon 6. In the beta form, the hydroxyl group is on the same side of the ring. So here in blue, the hydroxyl group in the alpha form, opposite sides of the ring. If we're looking at the beta, there's the hydroxyl groups here. They're on the same side of the ring. So here we're just looking at a little more detail for how the 6 carbon sugars form ring structures and the difference between those alpha and beta forms again. And again, the beta form has the OH group of carbon 1 on the same side of the ring as that hydroxyl group at position 6, while the alpha has it on the opposite side. This is showing those furanose ring structures coming and closing together. So again, the difference between those pyranose and the furanose structures is it an aldehyde or a ketone that's starting within that sugar? The pyranose would be the aldehyde, and the furanose would be the ketone that's present in the structure. Again, this is just another example. We're looking at the furanoses versus the pyranoses. And we are looking at the alpha versus the beta in each of these, just to further highlight those structural differences. So remember that in the alpha, you've got that OH group on opposite sides, and the beta, it's the same side. So there's same side. Here's our opposite sides. Because we know that these sugars, if they're greater than 5 carbons, like to form cyclic structures. Here we're just looking at those the 6 common monosaccharides that we've previously looked at in Fischer projections. We're just showing them here as cyclic closed ring structures. So that ribose, deoxyribose, glucose, fructose, galactose, and mannose. And I will add here that when we had the Fisher projections when they were straight, not closed ring structures, when you do form those ring structures, we're referring to these as Haworth projections. Now the pyranose rings, they can adopt two different conformations. We can have the boat form and the chair form. And this shouldn't be unfamiliar to those of you who have taken organic chemistry, which should be just about all of you. In the boat, there are substituents that have two orientations. We have the axial and the equatorial substances that are coming off. So you can see they're just denoted here with an A and an E for axial and equatorial carbons. Now this is looking at the boat and chair form of substances, conformations for glucose. And one thing that we need to consider within these structures is steric hindrance. And steric hindrance is going to help dictate which form or which conformation we actually see. Now, in this particular substance, in the glucose, that boat form, this form, is not going to be favored, and that's because it has greater steric hindrance. And that really just means you don't want a lot of those bulky groups really close to one another. Now, the furanose rings that we look at, they can also adopt multiple forms. Within these structures, they tend to pucker and form an envelope conformation. And we've seen these before when we've studied DNA or nucleotides. We see that there's the C3 endo and the C2 endo form of these structures. And that really just means that there's a particular carbon that is out of the plane relative to the other carbon molecules. So in the C3 endo, it's the C3 carbon, and in the C2 endo, it's the C2 carbon. Now, monosaccharides can actually be linked to other substances, and these linkages occur, or they're referred to as glycosidic linkages. And the glycosidic linkages come in two forms. You have an O glycosidic linkage and an N glycosidic linkage. The O glycosidic linkage involves an oxygen linker, and the N glycosidic linkage involves a nitrogen linker. And we see that here with A and B. Our glycosidic bond here, an O linker, and here we have an N linker, a nitrogen that's attaching these. Now these linkages can be alpha or beta, and that just depends on which side of the ring the linkage is going to occur on. So in the alpha form, the linkage is on the opposite side of the plane as that C2OH group, opposite side of the plane in our alpha structure. And in the beta form, the linkage is on the same side. Now, there are three common reactants when we're talking about these substances combining with the monosaccharides. We tend to see alcohols, amines, and phosphates that are going to join onto these. In the alcohols, the bond formed between the anomeric carbon of the carbohydrate and the oxygen of the alcohol would be that O glycosidic bond. This is predominant when carbohydrates link to form long polymers, when they attach to proteins, etc. With amines, your anomeric carbon linking to the nitrogen atom of the amine, and that's an N-glycosidic bond. And the phosphates, you have an addition of a phosphoryl group, which is a common modification. And we're going to see this as we move through our study. But these are just three different things that tend to interact with. But O and N glycosidic linkages are the more common linkages that join monosaccharides to other substances. So in addition to modification at the anomeric carbon, these sugars, these monosaccharides, can be modified at other positions other than that anomeric carbon. And that modification can be the attachment of functional groups to different places. So this is just showing a variety of different substituents that are going to replace hydroxyl groups in the overall structure. These types of modifications that we see here are usually going to be expressed on cell surfaces, so they are associated with the carbohydrates on the cell surfaces. But again, this is just to show examples of how we can have modification at other positions, and that modification can occur through different types of functional groups being added and replacing hydroxyl groups. So I did mention when we were talking about the modification that we can have phosphorylated sugars. So they can be phosphorylated. When that happens, it makes the sugar anionic. And that negative charge prevents the sugar from spontaneously leaving the cell. That negative charge is also going to prevent them from interacting with transporters of unmodified sugars. It creates reactive intermediates that can more readily undergo metabolism. So phosphorylated sugars will be key in energy generation and biosynthesis. And so just looking at the molecules that we have here, this glucose 6-phosphate that we see, this is the first step in breaking down glucose to obtain its energy. So we take that glucose and we trap it in the cell so that we can carry out more reactions with it. And we'll study this all in greater detail later. And then over here, this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These are intermediates in the metabolic pathway to break down the glucose. So phosphorylation is important. It makes sugars anionic. It prevents sugars from spontaneously leaving the cell across that lipid bilayer. It prevents interactions with transporters, and it creates reactive intermediates. So phosphorylation is pretty important for these monosaccharides. Now, there are some reactions that take place with monosaccharides, and we've just listed these reactions here. You can have oxidation to acidic sugars, reduction to sugar alcohols, glycoside formation, phosphate ester formation, and amino sugar formation. And for now, I just want you to know that there are different types of reactions, and these are the different types of reactions. We'll look at these in the context of other things that we study later on in the semester. So overview of monosaccharides. Here's looking at some of the monosaccharides and the common monosaccharides that we see here. And here are 4 listed and what they're important for. And then monosaccharide derivatives we talked about. You can have acidic sugars, so oxidation to acidic sugars, sugar alcohols that were obtained by reduction, glycosides, which are a reaction with an alcohol, phosphate esters, and amino sugars there. Now, individual monosaccharide substances can be linked together to form complex carbohydrates. So glycosidic bonds can join one monosaccharide to another monosaccharide. These complex carbohydrates will have directionality, and that's going to be defined by their reducing and non-reducing ends. The reducing end has a free anomeric carbon. Given that the monosaccharide may have, or does have, many hydroxyl groups, it means that many different glycosidic linkages are actually going to be possible. And so when we're describing the glycosidic linkages, we use the alpha or beta so that we can indicate what side that occurs on. And then we also describe it in terms of which carbons it's going to occur between. When we have 2 or more monosaccharides that are joined together, we call these disaccharides. They result from a glycosidic linkage. And some of the common disaccharides that we'll see include maltose, cellobiose, lactose, and sucrose. So here, the glycosidic linkage will form between a hemiacetal or hemiketal group of the sugar and the hydroxyl group of an alcohol, which is what we've talked about when we first introduced these reactions forming. Here, one monosaccharide will provide the hemiacetal or ketal. So this comes from the first, or one monosaccharide. And then the other monosaccharide will provide the alcohol group. So here we're just looking at some of those common disaccharides in more detail. We have maltose here. So our maltose. This is malt sugar. It's a disaccharide made from 2 D-glucose units. So these are both going to be D-glucose, and they're brought together through a 1,4 glycosidic linkage. Okay, so we're describing the type of linkage that occurs. The linkage occurs between carbon 1 of one monosaccharide and carbon 4 of the other. So we call this 1,4. Maltose is a disaccharide that we can easily digest. Another disaccharide listed here is this SOABiose. It's a disaccharide produced as an intermediate when the larger polysaccharide cellulose is broken up. This particular substance contains 2 D-glucose units, and these come together in a beta 1,4 linkage. So beta 1,4 linkage here. Humans can't digest this, the SOABiose, and that's because we lack the enzyme that's necessary to do so. We also have lactose here. There's our lactose. This is made up of beta D-galactose units and a D-glucose unit. So galactose and glucose coming together. When they join, they are going to form a beta 1,4 linkage. This is the compound that's found in milk, and this is a substance that we can digest unless you're lactose intolerant, and then you have some issues with digestion of that. And then lastly, we have our sucrose that's shown here. This is the most abundant of all disaccharides found in plants. It's table sugar. The structure forms through a glycosidic linkage between glucose and fructose. And here we see that it's a 1,2 glycosidic linkage that takes place. So this further illustrates that there are different ways that these structures can come together. They come together through different linkages between different carbons, and we can describe those when we're looking at the overall structure. So disaccharides are 2 units coming together. Oligosaccharides, you'll see the definition vary slightly, but typically these are carbohydrates that have somewhere between 3 and 10 monosaccharide units that are bonded together, and each unit is added to the next through a glycosidic linkage. Generally, these are present in association with other complex molecules. So raffinose, saccharose, these are just substances that represent oligosaccharides. So the raffinose, you have a galactose, a glucose, and a fructose unit, and this stachyose is 2 galactose, 1 glucose, 1 fructose. So here we have 3, here we have 4. But anything between 3 and 10 we tend to consider to be oligosaccharides. These are also the substances that are involved in the blood types, the oligosaccharides in general. So this is just showing blood types. You have your O, A, and B. The different symbols that are present here are just representing different sugars. So for example, the triangles that are here, these triangles are going to be fucose. These circles, these are galactose. The gold squares are N-acetylgalactosamine, and the blue squares are N-acetylglucosamine. So just different sugars coming together to form the overall structure that serves as our antigen for our blood groups. An oligosaccharide because it fits within those 3 to 10 units there. Then we can have these larger ordered structures, which we refer to as polysaccharides. So polysaccharides are polymers of many monosaccharide units which are bonded, and they will all come together through those glycosidic linkages. Now there are 2 types of polymers that we can describe in terms of these polysaccharides. We can have these homopolysaccharides or these heteropolysaccharides. So the homopolysaccharide just means that you have the same unit that's repeating within your overall structure. The hetero, you have different units that are present within that structure. Polysaccharides are not these sweet sugars that we think of. There are some tests that you can use for determining carbohydrates. Polysaccharides will not show positive tests for these, Toland's and Benedict's solution, and we may be able to do some of these in lab just depending on time. Monosaccharides are very sweet on the other hand, and they will show positive tests for some of these. Because polysaccharides are so large, they do have limited solubility in water. And just some examples of these polysaccharides that we may encounter. We have cellulose, glycogen, and these substances are storage polysaccharides. Chitin, which is a structural polysaccharide, and something like hyaluronic acid, which is an acidic polysaccharide. When we do have these structures coming together, we can actually have branching that occurs. So glycogen and starch are storage forms of glucose, and glucose is an important energy source for life. When high concentrations are present, we need to actually take this glucose and we need to store it. And we store it as a large polymer, and the reason that we store it as a larger polymer is to prevent osmotic imbalance within the cell. If we have free small glucose molecules, that is going to disrupt the osmotic balance. But if we have extra glucose and we store it in a large polymer, that's not going to impact the smaller molecules that are present, which would ultimately not disrupt osmotic balance. With glycogen, most of the glucose units are going to be linked by alpha-1,4 glycosidic bonds. Okay, so we have alpha-1,4 glycosidic bonds that are going to occur. That's going to be kind of in the individual chains, but every so often in the glycogen substance, the storage form of glucose, you end up with a branch. And the branches will be formed by 1,6 glycosidic linkages. Okay? So branches are going to be 1,6. Normal chains are going to be 1,4. And the branch points are going to be present about 1 in every 10 units of sugar. So glycogen, storage polymer, it's a homopolymer of glucose units. It's important because we don't want to disrupt osmotic balance. And it has 1,4 glycosidic bonds, alpha-1,4 glycosidic bonds, and then branch points are alpha-1,6 glycosidic bonds. And those branch points occur every 1 in 10 units. Here we're looking at cellulose and starch. Starch and glycogen are also often, considered, well, they're classified together. Starch is the storage polymer of glucose in plants, whereas glycogen, we're looking at animals here. So the starch is a homopolymer of that glucose, and there are two forms of that, amylose and amylopectin. Amylose is going to be an unbranched form, and it has alpha-1,4 linkages. The other is that amylopectin. This is a branched form of starch. So this, because it's a branched, you're going to have those alpha-1,4, but you're also going to have the alpha-1,6 linkages. And in the amylopectin, these branch points are going to be about 1 linkage per 30. Okay, so not as highly branched as the glycogen that we've seen. Now the cellulose that we see here, our cellulose is on the left. This is a major polysaccharide that's found in plants. It has structural roles. It's among the most abundant organic compounds in the biosphere. This is going to be unbranched. It has beta-1,4 linkages. That beta form allows for the formation of long, straight chains, which makes it good for that structural role. Now, we mentioned this before, but mammals cannot digest this because we lack the enzyme that allows us to do so. So this actually will serve as a great insoluble fiber. So polymers of monosaccharides are very important. Now, carbohydrates can be covalently attached to proteins as well, and these form glycoproteins. Approximately 50% of the proteome will consist of glycoproteins. So the proteome is all the proteins that exist. So about 50% of that has glycoproteins, or some sort of sugar attached to a protein. Generally speaking, glycosylation will greatly increase the complexity of the proteome. There are three main classes of glycoproteins that we study. We have the glycoproteins themselves, that's class 1. Proteins. We have proteoglycans. And we have mucins. The glycoproteins, in this case, the protein is the largest component by weight. Protein is largest component. When we're looking at the overall weight. Many are components of cell membranes. So glycoproteins, these are going to be components of cell membranes. Proteoglycans, the protein component is conjugated to a particular type of polysaccharide called a glycosaminoglycan. And here the carbohydrates make up a large percentage of weight. These proteoglycans are going to function as structural components and lubricants. And then we have the mucins, and the mucins are going to be mostly carbohydrate by mass, so predominantly carbohydrate by mass. The carbohydrate portion it's an amino sugar. And mucins are key components of mucus and lubricants. So this is just showing N-linked versus O-linked glycoproteins rather. So sugars are attached to the amide nitrogen or the oxygen atom that's present, depending on what they're attaching to. The N-linked oligosaccharides have a common pentasaccharide core, which is 3 monosugars and 2 N-acetylglucosamines, and then additional sugars can be attached to that overall core. In terms of glycosylation, this figure here is representing an important glycosylation reaction. We have N-acetylglucosamine, which is covalently attached to serine or threonine residues, and it's the serine or threonine residues of a cellular protein. The concentration of the glycosylated substance will reflect active metabolism of carbohydrates, amino acids, and fats. It indicates that nutrients are abundant. So this is just one specific example of glycosylation taking place. And this is just here to show that there's a variety of substances that form glycosaminoglycans. So there's diversity in structure. Looking at mucin structure, these substances are extensively glycosylated at serine and threonine residues by N-acetylgalactosamine. Mucins are capable of forming large polymeric structures. They're common in mucus secretion. They're abundant in saliva. So the general structure is shown here. There's a variable number of tandem repeat region that's present. It's rich in serine and threonine residues, and these are O-glycosylated. The carbohydrate portion of this overall structure can be as much as 80% of the overall weight. So it really truly is mostly carbohydrate by mass. These mucins will act as protective barriers in epithelial cells. They have roles in fertilization, in immune response and cell adhesion, and they're overexpressed when you have bronchitis or cystic fibrosis. So how does glycosylation take place, or where does it take place? Protein is synthesized by ribosomes and attached to the cytoplasmic face of the endoplasmic reticulum membrane. The peptide chain is inserted into the lumen of the endoplasmic reticulum. N-linked glycosylation begins in the ER and continues in the Golgi complex. O-linked glycosylation takes place exclusively in the Golgi complex. It does not begin in the ER. Now, we've been talking about glycoproteins, so proteins that have glycogen attached to them, but here we're looking at a class of molecules called lectins. Now, lectins are specific carbohydrate binding proteins. So they're not proteins with carbohydrates attached to them. They are proteins that can bind carbohydrates. They bind specific carbohydrate structures on neighboring cell surfaces. The partners for lectin binding are often the carbohydrate portion of a glycoprotein. So these proteins, these lectins, will find the carbohydrate portion of a glycoprotein and bind to it. And lectins are important because they facilitate cell-to-cell contact. They can be divided into different classes that are based on the amino acid sequence and the biochemical process that's involved. But one of the larger types that are studied is the C type, or calcium required, and that's what's shown here. These have a homologous domain, so all C type, of about 120 amino acids that's responsible for actually binding the carbohydrate. And this last slide is just showing us an application of where these types of molecules might be important. So many pathogens will gain entry into host cells by adhering to cell surface carbohydrates. So they can bind those cell surface carbohydrates and then gain access. Here we're using influenza as an example. The influenza virus recognizes sialic acid residues that are linked to galactose residues present on cell surface glycoproteins. So the glycoproteins on the surface of the cell, influenza is able to interact with that. So hemagglutinin is the viral protein that binds these sugars. After it binds, the virus is engulfed and it begins to replicate. And then the exit process is kind of reverse of the entry. But this shows us how these glycoproteins really are significant in terms of function of cells and kind of communication between cells, good and bad. So lots of information in this chapter. We've looked at carbohydrates overall, the different classes of carbohydrates, their structures. We've identified the types of reactions. We've looked at specific types of modifications, and we've looked at some applications.